Good morning, everyone. Okay, so welcome back. Just waiting a few seconds here to get everybody logged in. Okay. All right, so welcome back on this chilly, frosty morning. I hope you're doing okay. Okay, so we're just getting folks logged in here. Okay. All right, folks. Let's get started, okay? So I thought this morning it might be useful for us to talk about how your grade will be calculated in the course. We're just past lab exam 2 at this point, right? The average on that was roughly a 64%. So what I'd like to ask you to do is to log into the laboratory portion of Wiley Plus for me, okay? While we're just sitting here getting a few more people logged in, please log into the laboratory portion of Wiley Plus, because you're going to want to have access to your homework grades, okay? Okay, I think we'll get started here. I'm going to share my screen with you, and again, for those that are just getting logged in, we're going to start this morning by talking about how grades will be calculated at the end of the semester. And to do that, it would be helpful if you log into the laboratory portion of Wiley Plus so you can look at your homework grade, okay? All right, I'm going to share my screen with you. All right, and what I'm going to start with this morning is actually bringing up an Excel spreadsheet. Okay, so what I'm going to do is set up a hypothetical calculation about a grade, and what you can do along the way at your home is, observe what I'm doing. And if you have access to Microsoft Excel, or if you just want to write things down on a piece of paper to calculate your own personal grade, that would be quite okay, and I encourage you to do that. So, okay. So calculating your grade in the course is fairly easy if you have software like Microsoft Excel. If you don't have Excel, then it's a little bit more cumbersome, but not anything that's unreasonable if you have access to a calculator and pen and pencil, okay? So first off, I'm just going to create a couple little headings at the top of my worksheet here to tell you what it is I'm actually doing. All right, so we have 4 columns in this spreadsheet. Column A is going to be the name of the exam, such as lecture exam 1, lab exam 1, that kind of thing. The second column is going to be the grade that you earned on the exam. The third column is going to be the weight of the exam or homework, and that's taken from the front page of your syllabus. Okay, you don't have to bring that up. What I mean is you don't have to get it if it's out of sight. I'll provide it for you here today. And then we also have the weighted score, okay? And I'll explain what that means in just a moment. So, okay, in this course, you will be graded, your final course grade will be the cumulative effect of having 7 different things brought together, okay? So you're going to have lecture exam 1, lecture exam 2, lecture exam 3, lab exam 1, lab exam 2, lab exam 3, and then there's all your homework that you're doing on a weekly basis on Wiley Plus, okay? All right, so I'm not going to use anybody's specific individual grades. I would never do that. What I'm going to do is a hypothetical calculation where I put in some numbers, and to do the same calculation for your own individual personalized grade, you would obviously substitute your own exam scores in the calculation. Okay, so lecture exam 1, the class average was a 71%. So I'm going to use the class average as my grades here. Okay, so you're going to put your grade in as a decimal version of the percentage. So it was 71%, so that's 0.71. Okay. Lecture exam 2, we have not yet done, and lecture exam 3, we have not yet done. So I'm going to skip those momentarily and come back to it. Okay. Lab exam 1 you took, and the class average on that was a 68%. Again, wherever I put numbers in the grade column, you would substitute your own numbers in the grade column. Lab exam 2 you just finished, and the class average on that was a 64%. Okay. Lab exam 3 you haven't done yet. That's at the end of the semester. So there's a couple things we need to do. Okay, your homework. For those of you that have been able to log into the laboratory portion of Wiley Plus, there should be a grade tab along the left-hand side. Okay, a grade tab, grades. If you click on that, it should bring up a list of your homework grades for the various homework assignments you've done. Okay, so let me just ask you in the chat window. Do you see the grades tab in Wiley Plus? Okay. So I'm not going to show this on my screen. I'm not going to share my screen and show it to you because I can't do it without sharing private information, and I can't do that. That would not be acceptable. But what I want you to do is you should, on your screen where you're seeing your various assignment grades, there should be a scroll bar perhaps at the bottom where you can scroll the whole way over to the right. And there should be a column. There should be a final column on the right that says total. Okay. So question in the chat window, do you see the total column? Do you see the total column? Okay. The total column is the percentage, is your homework percentage of all the homework assignments you've completed so far, all the points you've earned versus the total points you could potentially have earned. Okay. So that total column is where you need to take that number. Okay. So obviously we haven't completed the semester yet, so that total column is going to change by the end of the semester. But right now it's a useful marker for how well you're doing with the homework assignments. So take your total percentage from the rightmost total column in Wiley Plus and put that down in the homework. So I'm going to put that down. I'm going to say that the percentage on that for this hypothetical calculation is a 92%. Many of you are doing very well with the homework. Okay, so we'll put that down as an example. Okay, any questions so far for what I've done? Okay, all right. So we have to deal with these boxes here that have no numbers in. And if I'm being honest with you, I'm going to turn them yellow so you can distinguish them from everything else. The boxes in yellow represent exam grades that you have yet to earn, right? Yet to earn because you haven't actually taken the exams yet. So for us to do this calculation here, we have to put some numbers in here, which in fact will be an assumption. Okay, so what I would like you to do just to complete this calculation is I want you to think about how the course is going for you as an individual. I want you to think about how it's going for you as an individual. And I would like you to make an estimate of how well you think you can do on lecture exam 2. Okay, think about how well the course is going for you. Think about how well you understand the current lecture material, and then make an educated guess about how well you think you may do on lecture exam 2. So I'm going to put in 75% for lecture exam 2 as my educated guess. You're going to have to do the exact same thing for lecture exam 3 because we haven't taken it yet. Okay, for lecture exam 3, I'm going to put in an educated guess of 80%. That's my educated guess. Okay, for lab exam 3, which is basically nervous system content, which we will start next week, you have to think about it. Well, okay, how well have the lab exams gone for me so far? You know, if I continue to really work hard on it, what do I think I can get on the 3rd lab exam? And so I'm going to put in an educated guess here of 70%. Okay, so everything in yellow right now is an educated guess because you haven't taken these exams yet. Okay, as you take the exams, those yellow blocks will turn to white because you will have completed the exam and you will know your exam scores. All right. So in terms of column C, the weight, I'm going to take the weights which are printed on the front page of your syllabus. So this was presented to you at the first day of class. Lecture exam 1 is weighted as 13% of your grades. You type in the decimal version of 13%, which is 0.13. Lecture exam 2 is 16%. Lecture exam 3 is 14%. Lab exam 1 is 13%. Lab exam 2 is 15%. Lab exam 3 is 15%. And your homework is weighted at 14%. Okay, so your homework is every bit as significant as lecture and lab exams. Okay, in other words, you are earning a part of your grade simply by completing your homework, and that's helpful to you in the final analysis. Okay, now to get the weighted score, what you're going to do is multiply the number in column B by the corresponding weight in column C. For example, this value right here will be equal to 0.71 times 0.13. Okay, that comes up to a weighted score of 0.09. You're going to do the same calculation for every grade and its corresponding weight and fill all these out. If you know how to set that up in Excel, I'll do it one more time for you. You can see what I did was I hit the equal button. I clicked on the number in the B column. I hit the asterisk sign on my keyboard for multiplication. I hit the 0.13 number in column C, and I hit return. Then I'm going to highlight that. And in the bottom right corner of that spreadsheet cell, there's a little square there. I'm going to grab that thing and I'm going to drag it right down. And the computer will understand that what I want to do is complete the same calculation for every corresponding row of the spreadsheet. Here we go. 1, 2, 3. Boom, there it is. Okay. So what you do at this point to get your final grade is to take these weighted scores and you add them up. So I'm going to hit the equal sign. I'm going to go up in the upper left corner, hit the sum option, SUM. That means it's going to add up all the numbers. So I'm going to take this and I'm going to tell it to add up all the numbers in the weighted score column by highlighting them. And I'm going to hit return or OK. And there, the final number of that calculation is 0.74. Okay, so 0.74 or 74.25% is your final grade in the course. Okay, again, this is just a hypothetical calculation. It doesn't reflect anybody's individual grades, but this is how your grade will be calculated at the end of the term. Okay, so do you have any questions? Okay, Rebecca, you're asking, we do have a final, correct? Rebecca, we do have a final, and the name of that final is Lecture Exam 3. Lecture Exam 3 is the final exam, okay? Lecture Exam 3 is the final exam, okay? It is scheduled for some time during the week of December 13th through 19th. I don't have the exact date yet for you. As soon as the administration releases that date, I will release it to you, okay? Okay, so do you have any questions about the process that was shared with you here today? This is how I will calculate your grade at the end of the semester. So if you understand this, now you know what you can do for the remainder of the term is every time you get a new exam grade, right, you can pop it into this calculation and update where your current grade is probably going to be with the remaining assumptions of the semester. Okay, so you can continue to track your progress for the remainder of the semester to see how well you're doing. Okay. Okay, so Danielle, you are asking me, can you go over again how to calculate the first number, please? Danielle, please tell me what you mean by the first number.
The lecture, like the grade and then the weight of the first number at the top.
Okay, well, okay, so the grades are what you earned on the actual exam, right? So like for lecture exam 1, you'd go into class marker and whatever grade was reported there, that's your exam grade. So that's where I'm getting those grades, right? Everything in column B for your lab and lecture exams come from your exams taken on class marker. The homework score is taken from Wiley Plus from the total column of your grades in Wiley Plus. The weights are taken from your syllabus. The front page of your syllabus tells you what these weights are. So these have been predefined weights that I set. I set the weight for each exam, and these are the weights that are being used to calculate your grades. Okay, so the weights are coming from the front page of your syllabus. The grades are primarily coming from your exams that you're taking on class marker.
Yes, I understand that part. I mean, like how, because I want to do it in Excel, so I'm trying to figure out how did you do the 0.71 and the 0.13 to get the 0.09.
Okay, so you would manually type in these numbers. So for the grade, I put in 0.71. For the weight, I put in 0.13. I manually type those in. And then for the next column, what you're going to do is hit the equal. If you're in column D2 like I am, you're going to select that cell. So it's selected, right? So now it's not selected. Now it is selected. Now it's not selected. Now it is selected. So I select it with my cursor, with my mouse. Then I hit the equal key. Then I select cell B2. I hit the multiplication key on my keyboard, which is the asterisk key. Then I select cell C2, and then I hit return on the keyboard. And that gives me the number.
Okay, thank you.
You're welcome. Okay, folks, any other questions? So, if this is something that you want to do, but you're struggling to do for yourself, let me know. What I mean is come visit me during office hours, or maybe grab me at the end of a lab session or something like that. I'd be happy to do this with you so that you know where you kind of stand in the course. Folks, I'm saying this to you because we are reaching a critical moment in the semester where you have to make a decision, okay? You have to decide whether you're going to complete the course and take the grade that you earn through all the exams and homework as your final grade, or you have to decide that you're going to withdraw from the course, take a W on your transcript and leave the course. Obviously, that's a difficult decision, a complicated decision. For some of you, where your exam grades have been consistently low, taking a W on your transcript is going to be a better option for you than taking a D or an F. Okay. So the deadline for withdrawing from the course, the deadline is, I'm gonna put this into the chat window. The deadline for withdrawing from the course is November 15th. Okay. So that's a little over a week away. And, you know, at this point, you have all the exams of the semester that you will have had by November 15th. Okay, we're not going to take another exam before November 15th. So you have all the scores you're ever going to have to make this decision. And so I want to encourage you to try to do this calculation. Think about where you probably are going to end up based on your educated guesses. And then you're going to have to decide whether you're going to see this through or whether you're going to leave the course. Okay. And I would be happy to talk with any of you individually in office hours about that decision. Okay. Okay. Any final questions before we leave this topic? Okay. All right, folks. So there you have it. If you want to chat with me at some point, you know, please come see me during office hours. I'd be happy to talk with you. Okay. So I'm going to get rid of that, and we're going to continue our conversation in chapter 10. By the way, let me ask this in the chat window. Here it comes. Have you located the Lecture Exam 2 study guide on Wiley Plus? Have you seen it? Okay. All right. So, you know, I just want to be sure you're aware that it exists, right? I don't want you to be going along and thinking, oh my goodness, I didn't receive a study guide. Okay. So it's there. Okay. So I'm seeing some doubt in the chat window. So hold on a second. Okay. So I'm going to start sharing my screen. Oh, I am sharing my screen, I think. Yes. Okay. And so I'm in the lecture portion of Wiley Plus. I'm going to the Files tab. Okay. Going to the Files tab. And if you go down and look around here, you're going to find Biology 121 Lecture Exam 2 Study Guide. Okay, so there it is, folks. I encourage you to download it, print it out, start thinking about it very seriously because your lecture exam is not too far away, right? Lecture Exam 2 is actually November 16th. So the day after you must make a decision about whether you're going to stay in the course or choose to exit the course. Okay, November 16th is Lecture Exam 2. And as you look across the study guide, you know, there are many things in this study guide that I've already spoken to you about during lecture time. I've provided support to you, but you're going to have some work of your own to do. Here's an example of something I have not talked to you about. Lambert-Eaton myasthenic syndrome. Okay, haven't talked to you about it. I'm not going to talk to you about it. In other words, this is an example of a challenge that I'm giving you because I want to see if you can, you know, if you can discover some knowledge on your own by doing a little bit of research on the web about this disease state called Lambert-Eaton myasthenic syndrome. Okay, so there's lots of things to start getting ready for in the upcoming exam. In addition to that, be aware that I've also placed a joint motion practice question sheet on Wiley Plus. Here it is. Okay. The idea is similar to the lecture exam 1 situation where you had different statements about tissues and you had to match the statement to a tissue category. Here I have different statements, which are the definitions of these various angular motion terms that we just went over last class. And I'm going to want you to match them up on the exam. Okay, so this is a practice sheet, and the answers are provided in an upside down version on the sheet. Okay, so any questions about that? All right, gang. All right, so hang in there. Let me know how I can support you. We're going to continue with chapter 10 here, okay? All right. So from chapter 10, we just got it started last time, right? We just got it started. In my textbook, I'm looking at the first couple pages of the chapter. In my book, which I have a physical printed version, is page 306 and 307. Let me put down some other ideas here. Categories of muscle tissue. Okay, this should not be a surprise to you because you have studied chapter 4. So we have skeletal muscle tissue, right? You're familiar with that. The skeletal muscle tissue is used to move the bones of the skeleton for the most part. There are some exceptions. For example, your tongue is a skeletal muscle, and it doesn't move a bone, it moves itself, right? So most skeletal muscle is used to move bones of the skeleton. Okay. Skeletal muscle tissue is striated. We pointed that out previously. Skeletal muscle tissue is voluntary. And what I mean by that is that you can consciously control its motion, right? I can control when my biceps brachii contracts to pull my palms towards my head. I can control that. And we'll say this, we'll say this, that skeletal muscle tissue, how it functions, how it behaves, what it does is controlled and regulated by both the nervous system and the endocrine systems. We'll have more to say about that later. Okay, truth be told, the primary physiology that we're going to share with you in chapter 10 is going to be skeletal muscle physiology. That doesn't mean the others aren't important. It just means we need to pick our battles right now because we have a limited amount of time. So cardiac muscle is the other category. You're going to get much more detailed conversation about cardiac muscle in AMP2. Okay, so cardiac muscle forms most of the mass of your heart wall. Okay, the tissue is striated. The muscle is involuntary, which is to say that your mind, largely your mind cannot control it, right? You cannot use your conscious mind to tell your heart to stop beating. And the cardiac tissue of the heart is controlled and regulated by both the nervous system and endocrine systems as well. The third type of muscle tissue that you know something about is the smooth muscle. Smooth muscle is found throughout our body. It's one of its most prominent locations is going to be throughout the tubing, of the gastrointestinal tract. Okay. Also going to be found in the walls of your blood vessels. Also going to be found around the hair follicles embedded in the dermal layer of your skin. Okay. Okay, so it's found throughout your body. Smooth muscle tissue is not striated. Smooth muscle tissue is involuntary. Okay, you cannot control it. And smooth muscle tissue is controlled by nervous and endocrine systems. All right. So we've told you last time about the major functions of the muscle tissue, right? Converting chemical energy into mechanical energy, also stabilizing body position, regulating organ volume, propelling food and liquid through the internal anatomy. Okay, so we've said that. Okay, it's in your notes. Let's talk a little bit about properties of muscle tissue. Okay. All right, so muscle tissue is electrically excitable. And that's a pretty amazing property. Basically, what that means is that your nervous system is capable of activating the muscle tissue to contract in a coordinated way. Okay, so muscle tissue is electrically excitable. Okay. Neurons have the ability to stimulate muscle contraction. Okay. In addition to that, I'm going to say some things that may not make too much sense yet because we don't have the background yet for it. Okay. Neurons have the ability to stimulate muscle contraction, and both neurons of the nervous system and muscle cells, muscle fibers, myocytes, they have the ability to not only receive electrical stimulation, but subsequently propagate electrical signals by generating their own electrical signals. These electrical signals are called action potentials. Both neurons and muscle cells have the ability to create electrical signals called action potentials. Okay. Generally speaking, like if I want to wave my arms around the room, if I want to do this, my nervous system is triggering the muscle contraction in my arms and shoulders to make that happen. So usually when skeletal muscles in particular, when they contract, it's because the nervous system is stimulating them. There are times, such as with your heart, where the heart sets its own contraction. It excites itself independently of the nervous system. And the only way to explain that is that muscle fiber sometimes can generate their own action potentials. It's kind of an amazing property of neurons and muscle cells to do this. We'll be talking much more about action potentials in the weeks ahead. Okay. Number 2, muscle tissue properties, contractility. Muscles have the ability to forcefully contract. I mean, that's their job. That's what muscles do, right, folks? That's their purpose in life. Okay, so muscles have the ability to contract, which means to shorten. Number 3, muscles are extensible. Extensible is a fancy way of saying muscle fibers have the ability to stretch, kind of like a rubber band can stretch. Now, obviously, you could stretch a rubber band to the point that it tears. You can also stretch a muscle fiber to the point that it tears. In fact, if you're exercising hard at the gym, maybe lifting weights that are just a little bit beyond your capacity, you're going to start to tear muscle fibers. And then overnight when you sleep, those muscle fibers are going to heal. And they're going to strengthen if you haven't done horrific damage to the muscle. Okay. So extensibility, muscles have the ability to stretch. And 4 is elasticity. Muscles have the ability to not only stretch, but then go back to their normal size. Going from stretching to back to normal size, that process, kind of like stretching a rubber band and then releasing the rubber band so it comes back to its normal size, that's called elasticity. Okay. Okay, so muscles are electrically excitable. They have the ability to contract, they have the ability to stretch, and they have the ability to stretch and return to normal size, which is known as elasticity. Okay. All right. I'm gonna start really rolling, folks. Lots of notes I got to get into here. So please slow me down with questions if I'm losing you or if I'm confusing you. We need to talk a little bit about the connective tissue that's associated with skeletal muscle tissue. I'm going to be looking at figure 10.1. Okay, a couple things to remember again. Once again, a muscle cell can also be called a muscle fiber, and it can also be called a myocyte. Those 3 ways of referring to muscle cells are synonyms. They mean the same thing. So a muscle cell equals a muscle fiber, which equals a myocyte. Okay. All right, we're going to be talking about various kinds of fascia that surround the muscle fibers themselves. So let me start off by reminding you what fascia actually is. Okay, fascia refers to sheets of dense, irregular connective tissue that lines body walls and surrounds muscles and other organs, typically for the purpose of stabilizing those organs, holding them into position so they don't inappropriately rub up against each other or collide with each other as you move, right? Because as you move, you know, your internal organs are shifting around a little bit. We don't want them to shift too much because they might damage themselves if they did that. So fascia refers to sheets of dense, irregular connective tissue that line body walls and surrounds muscles and other organs to stabilize them. Okay, so basically there are 3 layers of fascia that we need to talk about for skeletal muscle, and I'm going to be putting these into your notes and asking you to put an eyeball on them in figure 10.1 as we go through the list here. Fascia layers. Okay. All right. First, we have the epimysium. Take a look at the epimysium. If you're looking at figure 10.1 and you look at the rightmost column of terms, the term epimysium is shown to you as the third term down on the right. Okay, it's also the 5th term down. That doesn't mean it shows up in multiple locations. It means that the authors of the textbook are trying to show you epimysium from different viewing perspectives. So epimysium is the first layer of fascia. Okay. This is the outermost layer of fascia that encircles the entire muscle. So like if you could think about your biceps brachii in your arm, right, you would have a layer of epimysium around the entire mass of muscle that we call biceps brachii. This is the outermost layer of fascia that surrounds the entire muscle. Next, we have a deeper layer. This is the perimysium. Take a look. Perimysium is, in terms of the words on the right side of figure 10.1, it is the fourth term down on the right. The fourth term down on the right. It's also the seventh term down on the right. Okay, so the perimysium. Okay, this is the layer of fascia that surrounds bundles of muscle cells, or a group of muscle cells. A group of muscle cells has a special name. It's called a fascicle. Okay, so do you notice, again, looking carefully at the image of figure 10.1, there's this large bracket, a large bracket in the middle left of the image. It says muscle fascicle. Okay, muscle fascicle is a group of muscle cells. A group of muscle cells is going to have its own layering of fascia called the perimysium. This layer of fascia wraps around groups of muscle cells called fascicles. I'm going to highlight that word fascicle and make sure I get it spelled right here for you. Yes, Fascicle. Okay, so a group of muscle cells is called a fascicle. Okay. And then we have one more layer to talk about. We have the innermost layer of connective tissue, the innermost layer of fascia that wraps around individual muscles cells called the endomysium. The endomysium. So let me give you a starting point here. The term endomysium on the right side of figure 10.1 is the sixth term up from the bottom. Sixth term up from the bottom. Okay, the endomysium is the layer of fascia that surrounds each individual muscle cell. Okay. Now, what's significant that you appreciate at this point is the following. The epimysium is physically continuous with the perimysium, and the perimysium is physically continuous with the endomysium. By physically continuous, I mean that the perimysium is stitched to the epimysium. The endomysium is stitched to the perimysium. So let me give you an analogy and ask you to think about something. Imagine that you have a sweatshirt on like I do. You have your main shirt on like I do, and I have an undershirt on. I don't have an undershirt on, but imagine you had an undershirt on, a t-shirt on, okay? Imagine if somebody had literally stitched your undershirt to your outer shirt and then stitched your outer shirt to your sweatshirt. And so you were wearing all this clothing, and each layer of clothing was literally stitched to each other. So let me ask you something in the chat window. Here he comes. If your layers of shirts were stitched to each other and you pulled on the outer sweatshirt, would the inner layers also move? I mean, if they are stitched together, is everything going to move? hopefully you can see that the answer is yes. And that's the point. All these layers of connective tissue, which we're calling fascia, are stitched to each other so that when one layer gets tugged on by something, another layer is also going to get tugged on because they're all physically attached to each other. Okay. And so what I'll say here is I'm going to give you one more piece of information, and then we're going to do a crazy, well, let's see, we're not going to do a crazy thing yet because I haven't given you enough background knowledge, but pretty soon we're going to do a crazy thing. And let me say one more thing. So these layers of fascia that I'm highlighting on figure 10.1, all of them come together and merge at the anatomy of the tendon, which is connecting it to the bone, right? I mean, on figure 10.1, the second term down on the right side is tendon. So the tendon connects to the bone, right? And the tendon is connected to the epimysium. And the epimysium is connected to the paramysium. And the paramysium is connected to the endomysium, right? It's like that old song you learned as a kid, the knee bone's connected to the thigh bone, blah, blah, blah. Everything is stitched together. So the 3 layers of fascia listed above ultimately are continuous with the tendon that attaches skeletal muscle to bone. So you can imagine when the skeletal muscle contracts, it's going to tug on these layers of fascia, which are continuous with the tendon. So it's going to tug on the tendon. Since the tendon's connected to the bone's going to get tugged on, and the bone's most likely going to move in response. That's what I'm leading to here. Okay. All right, any questions so far? Before we go any further, we need to talk about some microscopic anatomy of muscle fiber. So I'm going to turn the page and take a look at figure 10.2. Okay. Let's see here. All right, you know that skeletal muscle fibers are multinucleated. We've said that before, and I think I've told you before, they're multinucleated because the primitive embryonic muscle fibers that were developing before you were ever born had individual nuclei, but there's tiny little muscle cells fused together like soap bubbles in a sink to build your mature muscle fibers. So each individual muscle cell contributed one nuclei into a single mature muscle fiber. So the single mature muscle fiber has multiple nuclei. Okay, you've heard me say that before. So what I'm going to do at this point is highlight some of these anatomical features here, and I'd ask you to follow along in figure 10.2 as we do so. All right, first we have the sarcolemma. Okay, folks, the sarcolemma is the fancy word we use to refer to the plasma membrane of a muscle cell. You know, from a beginning student's perspective, I think it would be very reasonable if you were thinking to yourself, why in the world do I need to call it the sarcolemma? Can't we just call it the plasma membrane and be done with it? Well, I get that kind of thinking, and I kind of agree with it. But here's the deal, right? The sarcolemma is the plasma membrane specifically of a muscle cell. The sarcolemma is not the plasma membrane of a neuron. You see what I mean? So as you become more educated, you got to learn more precise terminology, which refers to more precise objects. That's just how it is, folks. It's just the evil of the business we're in here, okay? So sarcolemma is the plasma membrane of a muscle cell. I'd like to draw your attention in figure 10.2 to the T tubules. T tubules Let me ask you a question in the chat window. Here we go. What color are the T tubules in figure 10.2? Okay, thank you, Faylin. Let's have at least one more person here. Okay, they're blue. All right, so hang with me here a second. So let me ask you another question. What color is the sarcolemma? Thanks, Faylin. Okay, good. Lauren, okay. It's blue. So think about it, folks. We just said the T tubule's blue and the sarcolemma's blue. Huh, maybe there's a connection here because they're both blue. There is a connection. The T tubules, which T is short for transverse tubules. Okay, so let's get this in your notes. The transverse or T tubules, okay, these are inwardly facing projections of the plasma membrane that penetrate deep into the interior volume of the skeletal muscle cell. The fancy word would be invagination. A T tubule is an invagination of the sarcolemma. It is an inward extension of the plasma membrane of the muscle cell. These T tubules are really, really important. Really important for these electrical signals we're going to be describing to you later on in the course, okay? So now we know what the T tubules are. Let's take a look at some other things. Sarcoplasm. Sarcoplasm is the muscle-specific term for the cytoplasm of the cell, okay? This is the cytoplasm of the muscle cell. And what I would bring to your attention at this time is a couple features that are unique and important that you appreciate about the sarcoplasm of a muscle, okay, of a muscle cell here. First off, the sarcoplasm is very abundant in storing glycogen. Okay, so glycogen is the animal tissue's primary storage form for polymerized glucose, right? Glycogen is polymerized glucose, which is to say I have glucose that are linked together with covalent bonds. So one glucose molecule linked to another glucose molecule, linked to another glucose molecule, a chain of glucose molecules. You might just think, well, that's odd. Why would that be inside the skeletal muscle? Well, think about it. The skeletal muscle needs to contract and it needs to move your arms. Think about it right now. You know, right now, many of you are probably sitting down, but as you stand up, as I'm standing up in front of you right now, I'm standing on one foot. I'm standing on one foot, which means the skeletal muscle fibers in the leg that's supporting my entire body need to be capable of supporting the weight of my entire body. And so that's going to require a lot of biochemical energy to do that. That's why skeletal muscle cells are loaded with biochemical fuel called glycogen. Okay, in addition to the glycogen, you should be aware of, let me ask you this question in the chat window. What is the name of the oxygen transport compound used in the bloodstream? What is it, folks? Starts with the letter H as in Howard. H as in Howard. Okay, Lauren, thank you. I'm going to run with your answer. It's hemoglobin. All right. So in the bloodstream, in the blood cells, there's a substance called hemoglobin, which binds oxygen, delivers it to your tissues. Folks, we're talking about muscles. We're not talking about blood. However, there's a related idea here. The sarcoplasm of muscle fibers are going to contain a chemical cousin to hemoglobin called myoglobin. Contains myoglobin, which is a chemical cousin to hemoglobin. Okay, and long story short, you can think of myoglobin as an oxygen battery. It binds to oxygen and releases it into the interior of the muscle cell when the muscle cell needs the oxygen. Okay. Let's see, if we have some electronics people out there in the audience, you might think of myoglobin as a capacitor. Okay, it stores a charge in a sense, and it releases that charge when the piece of equipment, the muscle cell, needs it. Okay, so myoglobin is a chemical cousin to hemoglobin. It stores oxygen and releases it to the muscle cell when needed. Okay, so we haven't talked in great deal about a process called aerobic cellular respiration. It was hinted at a little bit in chapter 3, I believe. You're going to get more of this in AMP2 next semester. But long story short, for your muscle cells to be so powerful, they need to be very energy efficient. The most efficient way of generating energy from sugar like glucose is to have oxygen available at the same time. That's why there's oxygen stored in the muscle cell attached to myoglobin, so you can efficiently extract energy from glucose. Okay? Let's continue. Number 4, myofibrils. Okay, myofibrils, just take a look at that. You can see the myofibril of figure 10.2. Let's see here. Probably the best image of the myofibril, arguably, would be panel C at the bottom. Look in the upper right corner of panel C and see the term myofibril. Folks, a myofibril is a contractile organelle. Okay, a contractile organelle. I have a question for you in the chat window. Here it comes. Hold on. Here we go. Is a myofibril inside or outside a muscle cell? Is a myofibril inside or outside? Okay. All right. As far as I can tell in the chat window, you are all spot on. You are correct. It is inside. Very good. Very good. Okay, so myofibrils are going to be instrumental in our understanding of the physiology, the mechanical portion of how muscles contract and relax. Okay. Keep that term in your mind and be aware of it. As we mature into adults, if we start doing weight training of some kind in the gym, our muscles are going to get bigger and we're going to get stronger, right? We're going to turn into Arnold Schwarzenegger, okay? The reality of it is, when we strength train like that, our muscles are not getting bigger because we're making more muscle cells. You don't get bigger muscles by making more muscle cells. The way you get bigger muscles is by taking the muscle cells you already have and physically making them larger. So each muscle cell gets larger because each muscle cell makes more myofibrils as you strength train. So you get stronger by literally creating more myofibrils, not more muscle cells. Okay. All right. Let's talk about some other things here. Next, the sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum, you can see there is colored orange, right? It's colored orange in the picture. Make sure you can identify that. Sarcoplasmic reticulum is a muscle-specific version of the endoplasmic reticulum. So hopefully you remember the phrase endoplasmic reticulum from chapter 3. At any rate, sarcoplasmic reticulum is the muscle version of endoplasmic reticulum. And what's important about the sarcoplasmic reticulum is it is a network of these, in the case of your textbook, color-coded channels. It's obviously not color-coded in a real cell, but it's a membrane, it's a network of membranes that's enclosed, it's self-sealed, like a balloon with a really odd shape. And inside the sarcoplasmic reticulum, your muscle fibers are going to store calcium. Calcium is stored in the sarcoplasmic reticulum and only released when the muscle cell receives an electrical signal from the nervous system. Okay. All right. So, you can see at this point in our conversation that we keep talking about this damn thing called calcium, don't we? Calcium keeps coming up, doesn't it? Okay. Calcium is deeply, intimately, pivotally important to the anatomy and physiology of your body. If your muscle cells don't have enough calcium, they're going to have to acquire calcium from the bloodstream. And if the bloodstream doesn't have enough calcium, it's going to have to resorb the calcium minerals from the extracellular matrix of your bone. And if you still don't have enough calcium, the only way you're going to get calcium is if you eat food and drink that has calcium. And the only way you're going to be able to absorb the calcium from your food and drink is if you have adequate supplies of vitamin D, right? So everything's really, really connected here. Okay, let's continue. Myofilaments. Myofilaments are chains of protein. So polymerized proteins that are linked together. And there's two types. There's two types of myofilaments you need to be aware of. There are actin myofilaments, which are sometimes referred to as the quote unquote thin filaments. And you have myosin myofilaments, which are quote unquote the thick filaments. Okay. Actin myofilaments are chains of actin protein. And myosin myofilaments are chains of myosin protein. Okay. Finally, there's one more thing I need to draw your attention to. and that is the sarcomere. Okay, in the bottom right corner of figure 10.2 panel C, bottom right corner, you should see a bracket there with the word sarcomere. Sarcomere. Folks, a sarcomere is a segment of a myofibril. Okay, a sarcomere is a segment of a myofibril. That segment of myofibril is made by overlapping actin and myosin myofilaments stacked on top of each other. Okay, so do you see that in the bottom right corner? You see the bracket sarcomere, and it's the same area of the picture that's also highlighting the myosin and actin filaments, and you notice the repeating pattern of how they overlap with each other. Okay. Okay, sarcomeres are referred to as contractile units. So I want to be very, very clear here about something. Okay, if we go up and look at number 4, myofibrils, we define a myofibril as a contractile organelle. Contractile organelle refers to myofibril. If we say the phrase contractile unit, we are referring to a sarcomere. Okay, it's just one of those things that you have to be familiar about the precise terminology used by scientists studying this topic. Contractile organelle is the entire myofibril. Contractile unit is a segment of the myofibril called a sarcomere. Okay, folks, any questions so far? So you can see, as with every chapter you've had to wrestle in this course, that there's a lot of new vocab, Okay, let's continue here. I need to highlight some specific muscle proteins in your notes because they're going to be important for our future conversation about the physiology of muscle. Okay, so I'm not going to give you an exhaustive list, but I'm going to talk about some major ones that are important to me and should be important to you. Okay, so I'm going to be referencing table 10.2, and again, I'm not going to list everything in that table in your notes, but I need to talk about some of them. Okay, so first off are the contractile proteins. And we've already listed them a little bit here. They are actin and myosin. Okay, actin and myosin are going to interact with each other in a team kind of relationship. They form a team with each other to help shorten the muscle fiber. Okay, so those are the contractile proteins. We already have those in your notes. There are proteins involved in regulating the contractile process itself. In other words, there are proteins whose sole function it is to turn the muscle contraction on or turn the muscle contraction off. And they're called tropomyosin and troponin. So tropomyosin and troponin work as a team to control whether the muscle contraction starts or stops. I will mention here that troponin as a protein has a binding site for calcium. So calcium is critical here in triggering the behavior and functionality of these two proteins, tropomyosin and troponin. Troponin has a binding site for calcium ions. Okay. And let me ask you a question in the chat window. Here it comes. What structure inside the muscle cell has the ability to release calcium ions so they can bind to troponin? What do we call that, folks? Because we just mentioned it. Okay, awesome. Good, good. I appreciate you following along. You're absolutely right, Sierra and Tony. It's the sarcoplasmic reticulum. Okay. All right, and let's continue here. There are some structural proteins that you can see in table 10.2. I'm not going to list all of them out, but there's two of them that I do want to talk about. And first I'll mention titin. Okay. Titin, the titin protein is named titin because it is so physically large. In fact, it is the largest protein in terms of length known to be encoded by your genes. It's the largest protein, folks. Okay. This is the largest protein known to exist in your cells. Titin as a protein. Now let's see if we can get a reasonable picture of it here someplace. Hold on a second. Yes. Take a look at figure 10.3. Take a look at figure 10.3. I have a question in chat window. What does a titin filament look like? What does a titin filament look like? Take a look at that. Give me some descriptive language in the chat window. What's it look like, folks? Okay, Lauren, you say a pigtail. I can understand that. Rebecca says a loop. I think that's what you're saying. Okay. All right. Okay, so not too bad. Folks, would you be okay with the idea that the titin filament looks like this in the chat window? It looks like a spring, doesn't it? Doesn't it look like a spring? Okay, that's what I want you to think about. Titin actually has the physical geometry of a metal spring. And so the point here is that titin can stretch out, which is part of the stretching or extensibility characteristic of muscle cells. And when necessary, it can compress back. Okay, so titin as a protein and then as a chain of protein called the titin filament provides a lot of the elasticity and stretchiness of muscle tissues. Okay. Okay, so it provides much of the elasticity and extensibility of the myofibrils. And then please let me finish this last statement here. We need to talk about dystrophin. Dystrophin is a protein that links the end of the myofibril to the sarcolemma. Okay, and so you can see dystrophin. You can see dystrophin graphically represented on figure 10.2, the last picture, panel D as in dog. Take a look at dystrophin. Tell me what color it is in panel D. What color is dystrophin? Okay, it's blue. Folks, dystrophin links the end of the myofibril to the sarcolemma. Here's the deal. Okay, when dystrophin is screwed up, you get the disease you've heard about called muscular dystrophy. When this protein is abnormal, you get muscular dystrophy. Okay. Quick little story. When people think muscular dystrophy, they might think about one specific disease. Muscular dystrophy is not a single disease, it is a family of diseases, because geneticists have identified over 50 different mutations in the dystrophin gene that produce 50 slightly different versions of the dystrophin protein, which give you 50 slightly different versions of muscular dystrophy. So fixing one person's muscular dystrophy does not necessarily mean you could fix a different person's muscular dystrophy, because you could have abnormalities caused by 50 different things. It's a really, really complicated disease. Okay. All right. Folks, any questions? Okay. All right. Thanks for your time and attention. Have a wonderful weekend. I'll see you next Tuesday, folks. Take care.